everybody?
Yes, sir.
Yes, sir. Yes, sir.
Okay. First of all, thank you for inviting me to talk about something that we have done over a period of time, and I can share this with students. As a professor, over a period of time, I have made time to encourage high school students to come into my lab, spend some time, learn something, whether they do mistakes or they get something out of it. I was always encouraging them to come, and I'm going to tell you a story about that. SIF Qatar asked me to come share my thoughts and ideas with you, and I'm really thankful to all of you for giving me this opportunity. And hopefully we will learn something new today. Let me share my screen and begin some earnest comments about what I want to tell you. As you can see from my very first slide, it is all about developing science ideas in research projects. And here I'm going to use a case study of the presence of a compound called lozone in henna. All ladies, especially young children, would love to have henna applied on their hands and really nice patterns and so on. And so this was a thought that came across from a student, and I'm going to talk about that. Let me, before get to talking about the case itself, ask you, do you understand what exactly is research? Research is really, any scientific phenomenon. It could be social sciences, it could be a hard science, anything for that matter, that can be investigated is really what research is all about. Now, if you're looking for finding a subject that you want to research about, the best thing is to develop a curiosity as to how things work around you, how things work around the world. So when you want to do that, I think, one could follow these kinds of ideas. You have to brainstorm ideas. If you have some idea within yourself, think about that idea in all different directions possible. Talk to others. Particularly, you can talk to your teachers, you can talk to your parents, you can talk to your friends, and test out your thoughts. Are they valid? When once you think that idea is valid, you choose a topic, a narrow topic on which you can start researching on. And it is important that topic is manageable, not something that is really, what shall I say, unmanageable or very ambitious. It should be really, really simple, and you begin there, then it can develop into an ambitious project like the one that I'm going to show you. But once you've identified the topic, you need to find some keywords so that you can search the literature and see what has been done about it. And when you read through this literature, you have to be flexible and open-minded. It is possible your idea that you're thinking is already completely done, but you will find something new, something different that you can work about. You have to define the topic and then review what is known about it through literature, and then formulate a research statement. It is sometimes also known as hypothesis. When once you have that hypothesis, the next thing to do is you have to develop a research statement to a researchable question. In other words, a research question. That's where we begin today's talk. Now, in 2008, I think almost 2009, a family came visiting me in my office at Minnesota State University in St. Cloud, Minnesota. And the parents were those of the student's name written here, Vijay Atle. Vijay Atle was a 10th grade student. The family had moved from Michigan State to Minnesota State, and Vijay was very interested in doing something in the lab. And so they brought him along with his sister, who was younger to him, was in 8th grade or so. And we had a little conversation, and then I asked him, what are you interested in? And so on, since I was always encouraging these high school students to come in and learn something. And I was, going to let him do something. When the conversation was almost ending, the sister of Vijay, was playing with her hands, and she showed these really bright, beautiful patterns, like the pictures that you see here. And quickly, Vijay asked me, Professor, do you know what makes that color come? And I think that is where that curiosity started for him. And instead of giving him an experiment that we were doing in the lab, I told him, maybe you should investigate what exactly, makes that color. Now, if you go around in India, this is a picture that I took on a roadside. They had put this henna heart advertisement, and you can see on the side, a young man is applying henna to a lady on the roadside. This is very, very common in India. And so I asked Vijay to investigate about this. So now here is the curiosity that Vijay started out with. And now he already had a research question in his mind, but it is a huge question. So now I asked him, let's narrow down to what can we do about it. So in other words, narrow down the broad area into something that is manageable that we can do. So here are two questions that we, through discussion, arrived at with Vijay. What chemical compound or compounds present in henna, the henna paste or the powder, that cause the skin coloring? And the second question is, what type of chemical reaction lead to skin coloring? So these were the two questions that we narrowed down to, and I sent off Vijay to the library, go find something about it, and he came back, sweating and profusely apologizing, telling me that I don't know what to tell you. I could not find anything. I don't know what to do. So what that tells is, I gave him the questions, I gave him the keywords. When he started, so much information started coming in front of him, and he didn't know how to decipher what needs to be done. So we sat down and started asking this question with some papers that he had collected, and asked, what is our question? Our question is the chemical compounds that are present in henna that cause the skin coloring. So by going through, one common theme that we found was that it would always come up with this molecule that is shown in front of you. I understand most of you are in 9th to 12th grade, and this is an organic molecule. You may not have seen these kinds of molecules before, but bear with me, I will come and give you some ideas. So the molecule that you see here is named as lawsone, and it is also called 4-hydroxynaphthoquinone, and it is abbreviated as HNQ, and this compound is present in the leaves and of the henna plant. So this is what we found out. So we answered the first question through literature search. The next question that was on our mind was, what type of chemical reactions lead to skin coloring? You see, I am a chemistry professor, I'm a biochemistry professor. I'm interested in what happens and how does this happen. So when we went through, we found the answer that lawsone reacts with proteins from the proteins from the skin or in the hair. Our hair is pure protein, you should know. And it forms this compound called Ship base. The Ship base, if you see in this molecule, if you give me a second, let me get my pen. You see this group right there that you see is called a carbonyl group, and sometimes it is referred to as a keto group as well. And this keto group can also be present in other compounds as well. So that will react with a nitrogen and replace that oxygen with nitrogen and makes this compound, and that nitrogen gets positively charged, and that positive charge distribution, it is what makes the color to appear. So this is what we learned. So now Vijay became very curious and he asked, can I do an experiment in the lab? And I said, yes, you can do. So before I get to his experiment, I want you to start thinking about when you search literature and start exploring your ideas, you not only are trying to answer the question that you created for yourself, now you're also thinking other questions that come about. Especially as a professor, when a student comes to me, I'm thinking not only about that question, how can I develop this further? So it helps researchers to identify their own questions, hypotheses, examine what previously researchers have done, provide a context to the type of work that you want to do, and also provides an overview of what is known today and what can we do in future. So this is what literature search does. Now, here are the questions that came to my mind when Vijay was trying to research and bring these papers to me, and we were talking about why is lawson present in the henna plant? What are its biological properties? You have to keep in mind, any living organism will not make a molecule that it does not need. It is all about energy. We are an energy dependent organism and we will not waste any energy. Just like that, plants are also living organisms and they will not waste their energy. So there is a good reason why henna plant is making this molecule. And the second thing is, what part of the protein reacts with lawson? What happens to the properties when lawson reacts with these proteins and the protein components? Can we prepare these compounds in the lab? Can we study their properties? What are the other possible applications for them? So like this, there were many, many questions that came up. And through this process, I'll show you how we answered some of them. And hopefully you will get to see them. Now, when once you have come up with an idea, it is very important that you assess, is this idea feasible? Is it practical? Can we do this? Do we have all the facilities? Do we have access to needed the materials? Do we have equipment? Do we have time? Do we have money? Do we have personnel? You see, so many things come to fore, and you need to decide on these things. So practical matters are very important that we consider whenever we try to implement an idea. So remember, Vijay asked me, I told him, yes, you can do an experiment. So I went to the lab and I gave him a little protein solution. I put some in water, gave it to him, and searched through our chemical inventory. And we had this lawson chemical in the lab. And I asked him, go ahead, mix it. Remember, our body temperature is 37 degrees Celsius. Let's set the temperature to 37 degrees Celsius, mix them. Let's see what happens. And you can see what happens right next to that in that beaker. It forms a nice red, almost brown, dark brown Ship base in the solution. And he was very excited and he forgot to wash the beaker and he left it on the side and he went home. By the time he came back about 48 hours later, that compound has almost plasticized, polymerized, and we could not even dissolve that. So now you have to think about that, what is that plasticization is all about. When you apply this coloring to the skin or to the hair, to color the hair, it really forms that polymer that sticks to the protein. So that's the outcome of that. So when Vijay did few more experiments, and he played around, and he wanted to develop this into a science project to present it in a science fair, and I was all fine with it. But he did something accidentally that caught my eye. What he did, he took a glass plate and he made an impression of his thumb. And you can barely see that impression on this glass plate. And then what he did, he put some protein solution on it, and he also put some Lawson solution on it and left it there for some time, and for about 3 hours or so. And notice on that thumb print, already there is reaction happening, and you can see that fingerprint is showing up. So this gave us an idea that, oh, I see, we could possibly use Lawson to develop fingerprints, identify people, and so on. So that is what happened with Vijay Atle's experiments. By then it was about 3 months, and Vijay's family got moved back to Michigan, and he left. I know after that he would keep in contact with me. He scored 1200 out of 1200 on SAT scores. He went on to Stanford University. He completed his bachelor's degree in engineering. I think he did it in computer engineering, and then I think he now works for Google. I'm not 100% sure. I think he works for a really good company. So that is the story about Vijay Atle. Now, about the same time, I got a fellowship to go to Nepal as a Fulbright fellow, and I forgot Vijay's project and left. And then I came back to Minnesota, and then it was time for me to move to Qatar. So this is about the time I started thinking about this project again. So let's quickly answer something for the students. What are proteins? How are they formed? Proteins are polymers. They're polymers of compounds known as amino acids. These amino acids really react with one another and form the long chains of amino acids linked to one another. So what is an amino acid? So if you see a molecule that has a carbon right there, and it is attached to this amino group, also known as ammonium group, and it is also attached to the COO group, also known as carboxylic group. And then there is this R group here. It can be different, and there is a H. Now, if we take our proteins, any organism, doesn't matter whether it is human, animals, or bacteria, anything, and you cleave them, chop them up into amino acids, you find 20 different amino acids. Now, how are they really different? Let me bring up this slide and show you quickly what are we talking really about. Let me get back to this. So remember, this is what I'm talking as amino acid. All amino acids, this part is common. And this R group is the one that is different. Now, if you see, I have shown the R groups in here, and I want you to look at these three in particular. Notice one has a OH group, the other has a SH group, and the other has a NH3 group. Those are important for our next explanation. So I'm going to continue. So the amino acids really react with one another. The amino group really reacts with that carbonyl. You release that OH, and it forms this bond, and we call that bond as a peptide bond, and sometimes we refer to that as an amide bond. So don't worry. Okay. So the amino acids, as you've seen here, the string of pearls, each of that pearl is an amino acid. They're linked through this linkage, and it turns into a protein, and it folds as you see in this corner. I'm moving some pictures here. So you can see the protein folds, and then these R groups that I was talking about, they are on the surface. That is key to keep in mind. So whenever we are looking at these proteins, what makes a protein functional is that R groups that we see. So now I'm going to talk about, can we make amino acids react with losome? The idea is, how does this color come from? What are those reactions? So I'm going to point out, these are Dr. Vandana Thotatil is our colleague in the Department of Chemistry here in Qatar University, and Sarah Darwish, Nihal Saharan, and Nadal Junaid were undergraduate students who wanted to do this project. And so this is where I revived Vijay Atle's experiments, and they wanted to do these experiments. So we took losome and we mixed with this amino acid called lysine. Remember that amino group that I showed you before, and we made this compound, we cooked it up, and it produces a really nice, bright colored compound. And we repeated this with cysteine, and notice how it produces dark brown to almost black colored compound. This is very important. I'll tell you in a quick second. And notice here, you take the serine, which is that OH group, and you mix that, you get this dark brown colored compound. Now, remember, we apply this henna on hair to make it dark black or maybe brown in color. What is in the hair? The hair is made of keratin protein, and that protein contains a lot of this amino acid, that SH group. That's the reason why if you take a string of hair and burn it, you get sulfur smell, and that's because of the presence of the sulfur. So you can see how that henna, in other words, the losome, when reacts with the cysteine, makes that black color. Okay, so that's one thing to keep in mind. The other thing to keep in mind is this serine. If you look at the skin proteins in particular, they contain serines in them, and that makes this brown colored compound. So now we answered two different things here. Remember, going back here, this brown color that forms on the hand versus the dark black color that happens in the hair is because of the type of amino acids present in those proteins. So that is the first ever question that we answered. Now, the next question is that, remember, Vijay Atle had done an experiment where he had taken his fingerprint, put it on a glass plate, reacted the protein and losome on it. Now, I was talking to a colleague in the department. His name is Yusuf Iji. He is a professor in our department. He's an organic chemist. And he told me he has published a paper on losome that I should look at. So in his paper, he published that losome is a good ionic sensor or a molecular sensor. Now, we demonstrated that in the lab. We took some losome solution in methanol. We added a little bit of sodium hydroxide. Notice how it changes to orange to dark red in color. So that suggests that losome itself changes color depending on what type of chemicals it's exposed to. So now our question is, the compounds we prepare using amino acids, the Schiff bases, are they also sensors? Okay, I'll tell you the importance of the sensors in a second. Okay, now we took one of these compounds that we prepared using this lysine amino acid, and we recorded the spectra for these. Now, what is this spectra all about? You know, substances absorb light. They absorb light differently because of the different types of groups present in them. And we call these groups as chromophores. Now, we took the lysine amino acid, recorded its spectra, see the red line here in the visible region, in the light that we can see, almost no absorption. We took lozone, recorded its spectra. It has a nice peak in the area below 350 nanometers, almost little or no absorption in the visible region. And when we prepare this compound, notice how nicely it produces that new peak, suggesting we have produced a new compound as well as it absorbs really strongly. And I showed you the color of the compound before. Now we added some ammonia into that compound, and notice what happens to the absorption. It jumps, it shoots up. So essentially demonstrating that this lozone amino acid compound we have produced is a molecular sensor. Now we should ask a question, why are these sensors important? One of the common poisons we find in food we come across, water we drink, is cyanide, and the formula for that cyanide is Cn minus. The cyanide is a very toxic compound. About 1 microgram is more than enough to kill a person. That's how poisonous this compound is. That's how potent it is. Now, so therefore, it is important that we test our environment, particularly the industrial effluents, the landfills that contaminated with cyanide, and many of the foods we eat also contains cyanide. A good example is shown here is cassava tubers. It is more like yams, but it is a little bit more harder. It is grown in India in all over the place, particularly in the African continent. Cassava is a very staple food. If you grow cassava in very highly irrigated areas, it doesn't produce cyanide. You grow that in very arid conditions when the water content is very low, it has lots of cyanide in it. So people could easily die by eating cassava depending on how it is grown. So we tested some of these compounds. I'm showing only one example here to see if we can detect cyanide in particular, and the answer is yes. You can see that it changes color very quickly, indicates the presence of cyanide. Okay, so should I pause here? Anybody has any questions, or should I continue?
You can continue, sir.
Okay.
We'll take the question at the end. Yeah.
Okay, good. Okay, so now let's see, can we use these compounds for biometric analysis? This was the work done by these undergraduate students that I have listed here. And let me quickly talk about fingerprints. Fingerprints are the most commonly used biometric tool for identification of individuals. It is very unique to each person, especially when there is a mass disaster. To identify people, it becomes very, very difficult. What part belongs to which person and so on. One of the things that people use is fingerprints and DNA analysis. And think about crimes that are committed by people. People that when they go to rob somebody or steal something, they touch surfaces and leave behind their fingerprints. And this is so unique that the chances there is another individual that has the same fingerprint is about 1 out of 64 billion. Keep in mind, in the whole world, we only have about 7.5 billion people or so. So that means there is no chance there is another person that has the same fingerprint. I want you to keep in mind the identical twins share the same DNA, but their fingerprints are really different. So keep this in mind. All right, so how do these fingerprints come about? If you look at our skin, there are 2 layers, what is called dermis, the lower layer, and what is called the epidermis that is attached to that. If you look at the dermis in particular, it has these peg-like structures, almost like spikes, to which the epidermis gets attached. These pet-like structures are also known as dermal papillae. They're formed at the time of birth, or actually fetal stage, and they remain exactly the same throughout our life. The epidermis, when it gets attached to these pegs, it creates those folds, and those folds are really important for gripping surfaces and so on. So those really are the fingerprints. So the skin friction ridges that originate from the skin foldings is really the fingerprints that we come across. Okay, so when you touch a surface, you leave behind a fingerprint. Where does this come from? Look at your hands. Your hands are always moist. So that means we secrete, substances out of it. There is sweat coming out. There is also oils coming out. There are two types of glands in our skin. There are the sebaceous glands, and there are these oil glands that produce these secretions. And if you analyze the secretions, you find amino acids, and you also find proteins. The reason I brought this up is, can we use Lawson to detect fingerprints? So that means you can make reactions with these compounds. Okay, so remember that we are going to see if we can use the Lawson and its derivatives to identify people, and we are going to exploit the idea that the Lawson as well as the Lawson amino acid ship bases are chemical sensors. So this is the idea that we are going to utilize. Okay, so I asked the students to do an experiment. They would take a little glass plate and make an impression, add Lawson first, and then add some amino acid on top of it, and then you heat it for about 3 minutes in a microwave, a regular microwave that you use at home. And they did. So here is A, just Lawson alone, nice yellowish to orange in color, and you add amino acids. Amino acids are colorless, white powder, and when it covers, you can see that yellow color is somewhat going away, and you heat it, and it changes color, and then you expose it to a little ammonia, it becomes bright red. Very nice. So we repeated this with another compound. Notice here, this compound, this amino acid really react very quickly, and when you add ammonia, you can see how the color becomes really dark. And it is yet another example of going and enhancing the color. So what does this lead us to? It leads us to believe that these compounds can be easily used to visualize the fingerprints on surfaces that are not visible. Now, we also noticed that these compounds, if you expose them to UV light, the blue light, they start fluorescing. That is yet another characteristic that can be exploited to identify people who may have committed crimes, or people that left behind fingerprints for no good reason. Okay, so that is how we discovered this. I'm looking at my clock. I promise not to cross 50 minutes. Okay, so next we asked this. So we apply henna, and it forms a nice color. After 2 weeks, it disappears. Now, does this compound in any way provide any kind of protection to the surface that it is applied? So we asked this question, and I asked the students to explore this possibility. They did a very simple experiment. They took some bacteria, cultured it on a plate. You can see 3 different plates, and took a little filter paper wick on which they put one of this Lawson serine ship base, and they took a positive control. Here it is a real antibiotic, ampicillin. They put it on that filter paper wick, and they took Lawson by itself, put it on that filter paper wick, and left the bacteria to grow. So what you see is, look around the positive control, there is no bacteria growing around it. That shows that ampicillin kills bacteria. Now, if we did that with low zone, notice the area is getting clear, that it is also killing bacteria. And now come to low zone serine amino acid ship base, even that prevents the growth of bacteria. So in essence, applying Mehendi is kind of providing antibiotic protection to the surface and not let the grow bacteria on our skin. So that is what came out of this question, that do they have biological properties? Now I'm going to go towards the next part where we want to explore, can we use the ship bases and low zone to prevent Alzheimer's disease? So what is Alzheimer's disease? Alzheimer's disease is all about, developing this forgetfulness. Students, I want you to think about this. When you go visit your grandparents, they always complain several things. One is they have these knee and joint pains and they forget a lot. Forgetfulness is really related to Alzheimer's disease. Now, this work was done by Radhika Menon, Dr. Vandana Totattil, Mariam Jama, and Muhammad Yusuf. And here, I want you to start thinking about the question that we have is, can low zone and its ship bases modulate amyloid dynamics? I'll tell you, amyloid dynamics here really is all about neurological diseases or Alzheimer's disease. That's all I want you to think about. Now, before I get any further, I want to remind you back. I told you how the proteins are formed. Amino acids react with one another, form that peptide bond, and that continues and forms this long chain of amino acids linked to one another, which then folds and produces this folded protein. Folded protein in the correct conformation is most important for its function. So here is an unfolded protein, here is the folded protein. Folded protein is functional, unfolded protein is not functional. I want you to think about this for a quick second. I will give you an example. When you have fever, you feel miserable. Why do you think you feel miserable? You feel miserable because the body temperature goes up. What happens when body temperature goes up? When the body temperature goes up, the proteins in our body don't function normally. Some of them get denatured. In other words, they get unfolded. So, folding of proteins in the correct conformation is the most important thing for the function of any protein. So keep that in mind. So that means misfolding, unfolding is a problem. So we asked this question, are there diseases that are caused by misfolding of proteins? The answer is yes. Alzheimer's disease is one, Parkinson's disease is another one, cystic fibrosis, diabetes, many cancers are caused because of misfolding of proteins. That misfolding could be because there are mutations in genes and so on. We will not go into that part. So remember, misfolding of proteins is a problem. Okay, so let's try quickly understand what is this Alzheimer's disease is all about. Now, if we take a person that has died because of Alzheimer's, their brain, take a cross section and stain it for protein, you find these patches, these black patches. If you analyze those patches, it turns out it is protein aggregates, protein precipitate. Now, these protein precipitates, when produced, the neuronal cells cannot communicate with one another in the brain, and that is really what causes forgetfulness. And letting that could be really serious. Now, what are these proteins? These proteins are referred to as A-beta amyloids. Let me take 1 minute and explain this. We have this protein called amyloid precursor protein, also known as APP. And this protein is broken down into this beta secretase, gamma secretase that are very important in nerve growth growth of the nerve cells. And in the process of cutting that protein, you release these short protein peptides called A-beta monomers. These A-beta monomers can form, when produced in large quantities, oligomers. They can form fibrils, or they can form these aggregates also known as plaques. So that is really how these plaques are produced. And then those plaques are produced, the nerve cells cannot communicate with one another. And that is what leads to the forgetfulness. Now, I told you that misfolding of proteins is a problem. And I also told you there are many other diseases where misfolding of proteins lead to diseases. What can cause this misfolding of proteins? Environmental stress is one, mutations in genes is another one, and then how these genes are ultimately converted to proteins called translational errors is another reason. Now you have a protein that is produced which forms this intermediate fold which gets partially unfolded, and that partially unfolded one will start polymerizing, and that's what forms this fibrillar aggregates or plaques. So remember, I told you that there are many diseases caused by misfolding of proteins. So I asked the postdoc in our lab, Dr. Radhika Menon, to see if we can search through databases of proteins and find what is a common sequence in the protein that could possibly form these oligomers, fibrils, and plaques. And she came up with these that are shown here. They are simply each letter representing an amino acid, and it's a short sequence of the protein. Now, we need to be able to monitor, will they aggregate and form those plaques? So for this, we figured out there is an experiment we can do using this compound called thioflavin. Thioflavin binds to these aggregates of proteins and it fluoresces. You can see with time, the fluorescence increases with this amyloid peptide that I was talking about. And we asked, will these peptides that we have synthesized will do the same thing? Not as good as A-beta-42, but it does show that it can bind. But there are some which don't bind at all. So we had an idea of how we can do. Then the question is there a better way to do this? Can we see these aggregates under a microscope was our question. The microscopes that you've seen in high schools and other places are really low magnification microscopes. We cannot really use them to see molecules. But there are microscopes, scanning electron microscope, tunneling electron microscope, atomic force microscope are some of the microscopes that are available that we can see particles that are nanometer in length. Now there is, literature published. If you see, if you have oligomers, if you look under the scanning electron microscope, that's how they would look like. And here are fibrils, you see those dots kind of forming filaments, and you can see mature fibrils, and then you can see plaques that are produced. So now we asked, can we take the peptides that we found in these various diseases to be commonly present, that is these peptides that we synthesized, can they make fibrils? So we tested it with this thioflavin assay. There is our amyloid peptide, that's a positive control, and all these peptides that we synthesize do absorb but not very high. If you go with this data to publish something, people will laugh at you. They could tell you that is this simply background noise. You should really do better experiments. That's exactly what we did. We took the A beta peptide and through tunneling electron microscopy, looked for the formation of these fibrils.
The distance that you are traveling, the total time that you spend traveling and in consult, and based on that you can make how much are those charge, so that you can decide
Somebody's mic is open. Can you hide it, please?
Dear participants, please mute yourself.
Okay. All right.
Thank
So
you.
you can see now these peptides we created also produce really nice fibrils. Oops, let's get back. And you can see some of them produce this flax themselves, and you can see those lines, they're really fibrils, and various different types of structures are produced. So essentially from this, what I want you to take home is that misfolding of proteins can lead to diseases. And if you screen through the databases, you can find common peptides that are present in proteins that cause these diseases, and these peptides can aggregate and produce these plaques and fibrils and so on. So what's our next step? Our next step is, remember, we are really still dealing with low zone and low zone compounds, the ship bases. Can they prevent the aggregation and formation of these plaques in the lab? And so what we did, I'm going to skip this and go straight to this picture here. Here is the control experiment where we did not add the low zone to that. Very nice peptides. And you add some low zone to that, notice the peptide, those plaques and the fibrils are disappearing. And in this one, you can almost see completely disappear. So in essence, for the first time, we have shown that low zone is having anti-Alzheimer's properties. Now, we took that to apply to these peptides we created that can originate in other diseases or key proteins in other diseases. Here is one of them, very nice rods, and you add some low zone to that. Those rods really disappear or dissociate. You can see here in this case as well, those are disappearing when you treat it with HNQ. And here is another peptide, and you can see how they form these plaques. The plaques are really slowly dissolving. So clearly, we now demonstrated that the amino acid low zone by itself can act as an anti-Alzheimer's compound. Now, what we have not done is, actually we have done, but I don't have the data to present clearly in front of you, is taking one of these amino acid low zone ship base and see if we can dissolve these plaques. These experiments are ongoing and we are working on them right now. So let me conclude here so I don't make you get too bored. So clearly, research is something that comes out of passion. You have to have that curiosity, and you have to have time on your hand, and you need a lot of patience and resources. If you noticed, the number of people involved in doing all these experiments, there were a lot of different people and lots of amount of time spent on this. And so research is not something that you can do it like that, but you need patience. It takes time. You have to have that curiosity. So I showed you that low zone that comes from henna plant is really a novel compound. And when you combine that with low zone, amino acids and create those ship bases, they themselves are novel compounds. Not only Lawson is a molecular sensor, even the ship bases we create are molecular sensors. Some of them are really unique. The example that I showed you, very specific for cyanide detection. Now, we also showed that we can exploit this idea of sensing properties to visualize latent fingerprints. Latent means invisible fingerprints. And I showed you that these compounds has antimicrobial activities. Recently, one of the students has done some work where we show that these also have antioxidant properties. And I showed you that we can search through a protein data bank and identify diseases, the key proteins in diseases that may have sequences that could produce amyloids, in other words, those flac-like structures. And we can inhibit the formation of those amyloids. And this is potentially a therapy that could be developed for not only Alzheimer's, but other neurological diseases and possibly even diseases such as diabetes. So this story is another one, but I'm not going to talk a lot about this. I just want to mention there was another high school student that came to my lab when I was in Minnesota. Her name was Elizabeth Kreckelberg. She was in 12th grade at the time. And she came and did some experiments on an anti-cancer drug that's coming from this plant called Otilia. And ultimately, she presented her work to Intel Corp and earned a scholarship to go on to college. And she went to Columbia University and got her 4-year degree. So let me acknowledge the people who made all this possible. The support for doing all this work came from Qatar Foundation through National Priorities Research Program funding, from undergraduate research experience program. I also had some money coming from Qatar University. And you can see we have an organic chemist in the department, Dr. Yusuf Hiji, who helped us in advising creating the ship bases. And Dr. Radhika Menon did all the peptide work that you saw. The synthesis was all supervised by Vandana. And you can see the large number of undergraduate students that participated in this project. So I hope you enjoyed my talk, and I'm happy to answer if there are any questions. Let me stop here. And thank you for the opportunity to present this work to you.
Thank you, Doctor. Thank you, Dr. Lakshman. It's wonderful presentation. Thanks for enlightening us how a Lawson found in Hina can be put to various applications with the great research work done by you and your team. Now I request Mr. Ravi Kumar to take up the questions from the participants, please. Over to Mr. Ravi Kumar.
Thank you, Srinivas. Question number 1 that was asked in a very beginning stage from a student was, what makes a good topic?
What makes a good topic? Actually, what makes a good topic is simply what you are interested in. Think about it. Why are you asking that question? Because you've seen something that you don't understand. And so you want to research about it. That in itself is a good topic. But then one way to select a good topic is talk. Talk to others. Talk to your teachers. Talk to your parents. They could, give you ideas and explain what is going on with the phenomenon that you are looking at, and possibly give you even further ideas to explore, narrow down your question.
A question that is asked just now from Aujas Mehta is, why we will use Lawson if there is already a chemical named fluorescein which reveals the fingerprint much easily?
Very good. Good question. You really have to think about what surfaces you find fingerprints on, what kind of reagents are available. Fluorescein, is a physical application on a surface that sticks to what is on surface. Now, if you leave the fingerprints to stay there, water evaporates, lipids get degraded, and you apply fluorescein, it won't stick to that. So you need to be able to do some sort of chemical reactions to react with minor components that are present in old fingerprints. So there is one reason. The second reason is that not all compounds are suitable for all surfaces. Now, if you look at Lozone in particular, it can work both on porous and non-porous surfaces. Fluorescein really works well on solid, non-porous surfaces. It doesn't work on paper and other types where it doesn't react. So that's a good reason, I would say, why we should have new reagents.
The other question is ampicillin used to kill bacteria?
Yes, penicillin, ampicillin belong in the same family. Penicillin is used as an antibiotic for treatment. You think about amoxicillin and other antibiotics that are sold. Ampicillin is one such antibiotic. Kills bacteria.
Can you mute, please, Mr. Navaneet? Okay, so I have one final question, which is, how is the cyanide content tested in industrial effluents? Is it done in Qatar?
The answer is yes, it can be tested. What they right now do is they have developed strips which contain a molecular sensor on it that you can dip in water and take it out. It changes color depending on the concentration. So that's one way of doing it. The other way of doing it is they do some chromatography type of experiments where if you pass through particularly capillary electrophoresis, because it's a small ion, if it's present, it will produce a specific peak at a certain time, what we call retention time. So you can identify the presence of it. And it is not just in Qatar, it is done everywhere. You cannot release wastewater into large water bodies unless it is tested and the concentrations of poisons in particular, including cyanide, should be below certain threshold. Unless that happens, they cannot release it. So they do test the presence of it.
Thank you. I have a question that has come just in. Does Lawson has antifungal properties?
The answer is yes and no. I say yes because I have read some literature where somebody has claimed that it has antifungal properties, but I've not really seen the real evidence that shows concretely that it really kills. So therefore, I would want to turn on the safer side by saying yes and no, but one could do some experiments and demonstrate it very easily.
One more has come in. The compounds in henna have antimicrobial properties. So are they used in healthcare as of now?
As of now, I think they're used in folk medicine, Ayurveda and other places. They mix this henna plant and prepare some extracts for using them as antioxidants, but not in English medicine as a treatment.
Thank you. We now end up with a final question, which is, can Qatar University extend support for aspiring school students to take some experiments for validating ideas and expert guidance?
Actually, we always encourage students to talk to professors, and we welcome them and come do some things. But what we have noticed is unusually different than my experience of working in the United States for a long time. The students that we get here, they tend to think or expect that the professor do everything. So my caution is, when you approach a professor, you really should approach a professor that you're interested, you will do these experiments in the lab, and you really want to learn. And yes, it's possible to do it.
Thank you so much. I will
Professor
now hand over to
Lakshmi,
the moderator. Yeah.
Professor Lakshmi, I'll just rephrase the last question and ask for the sake of the student that do Qatar University allow the non-Qatari students to work in the university labs or approach to the professors?
I think you always approach the professors. You can also approach the department heads through the schools. And we always bring students from high schools to come see our labs and see how we do things and so on. So why not? Students can come and do these things. I had a set of students come from one of the public schools. Unfortunately, I'm not fluent in Arabic, and I could not communicate with them very well, and they did not know very good English. And so finally, I had to tell their teacher to be present when the students do experiments. Their teacher did not have time, so we could not take it to the next level. So it is possible, yes.
Thank you.
Thank you, Doctor. Thank you, Ravikumar ji. Thanks for the questions. We have with us Mr. Samir Karmakar ji, who is a scientist in Central Food Lab, MOPH. I would now request Mr. Samir Karmakar to give the closing remark on the session, please. Over to Mr. Samir.
Thank you, Mr. Srinivas, to join this meet. Thank you for giving this opportunity to speak something in this wonderful meeting. First, I would like to thank Dr. Lakshmina Sirana, Professor, Qatar University. This is my great pleasure to express my word of thanks. For students also, I would like, this is the wonderful opportunity for them to come forward and work something in the area of science. What I find in Qatar, and when I compare it to the Indian schools and universities, here we have a lot of infrastructure in terms of knowledge and in terms of availability of the machinery, what we have. And I would like or encourage our students to get that opportunity, use this opportunity and do some research work and make proud your parents, your teachers and our nation. Thank you very much.
Thank you very much. Thank you, Samir ji. Now I request Ms. Sailaja ji to present the vote of thanks.
Thank you, Srinivas ji. Good evening, everyone. Research is crucial for all academic and scientific pursuits, and the aptitude to conduct research is one of the most important skills a person could develop. On behalf of SIF Qatar, I would like to thank Dr. Lakshmaiah Sriramaji for providing insights on developing a thought or concept into research skills. Thank you, sir. We would like to thank Dr. Arvind Ranade, who accepted our invitation to attend this event today. Thank you, sir. I would also like to thank the teachers and the students and all the others present today for coming forward and joining the webinar, which definitely has many takeaways. We would also like to thank Qatar Press for the regular support they provide in publishing SIFQ activities. Last but not the least, I would like to thank SIFQ team for putting up things and organizing such an interesting event. Thank you all.
Thank you, ma'am. Thank you.
Thank you, Dr. Lakshmiya Sriramaji. It was
You are welcome. So we are done for the day.
Yes.
The
The
formal discussion has been over, so I am stopping the
Okay.
recording,
yes.
and then we can continue with the informal discussion. And I would like to ask the students if they don't have any informal question, then they can leave the meeting.